Let's begin with a quick recap. Recall that DNA, which is located in the nucleus, is transcribed to mRNA, which is then translated to a protein at the ribosome. The proteins produced here are vital to the functioning of our bodies. But what happens when the segment of DNA that codes for a protein is faulty or missing? If a segment of DNA is messed up, then the functional protein it codes for cannot be made. The absence of this important protein can result in disease. This is where gene therapy comes in. In gene therapy, scientists attempt to introduce a functional copy of a gene into the cells of a patient. But how do we get the functional copy of the gene into the patient's cells? The most common technique is through the use of viruses.
But Mr. Cannon, I thought viruses were bad.
Well, yes, Bobby. Traditionally, viruses infect cells, making a person sick. But in gene therapy, most of the harmful genetic information is removed and replaced with the functional gene that the patient needs. When a virus is genetically engineered to carry a therapeutic gene, it is called a viral vector. Today, we're going to focus on a specific family of viral vectors called retroviral vectors. Retroviral vectors store their genetic information in the form of RNA. That's important. Okay, let's look at the basic structure of a retroviral vector. First, there's an outer membrane, which is covered in glycoprotein receptors. Inside, there's a capsid, which is basically just an inner compartment. Inside the capsid, there are a couple strands of RNA. There are also a few enzymes, including reverse transcriptase and integrase. Alright, now it's time to put this vector into action. Let's say we have some retroviral vectors that have been modified in the lab to carry a gene that a patient needs. Then, we will take a sample of cells from the patient and put it into culture. Next, we will inject some retroviral vectors into the culture. Now the retroviral vectors can infect the patient's cells and introduce a functional copy of the gene into their DNA. But how? Let's zoom in to find out. Here is a retroviral vector approaching a target cell. When it gets close enough, one of its glycoprotein receptors binds to a protein receptor on the target cell, initiating the fusion of the two membranes. The viral capsid now enters the cytoplasm, where it breaks down, releasing the RNA and enzymes. Now it's time for gene integration.
Now wait a second. The viral genes are in the form of RNA, right?
That's correct.
And the host cell genes are in the form of DNA, right?
Mm-hmm.
So how do you fit the RNA into the DNA?
You don't. The RNA must be transformed into DNA in a process called reverse transcription. Do you remember what happens in regular transcription?
Yeah, RNA is transcribed from a DNA template, right?
Yup. So what's the opposite of that?
DNA is transcribed from an RNA template?
Yup, you got it. That's reverse transcription, and it's carried out by reverse transcriptase. Reverse transcriptase takes the viral RNA, shown here in red, and reverse transcribes it, using DNA nucleotides present in the cytoplasm. This new DNA strand is then transcribed again into double-stranded DNA. The viral genetic material is now in the same form as the host cell genetic material, but to incorporate this DNA into the DNA of the host cell, we need another enzyme, integrase. We are now inside the nucleus, where a segment of host cell DNA is shown. Integrase brings the viral DNA into the nucleus through a nuclear pore. Integrase now essentially cuts the host cell DNA, allowing the viral DNA to be integrated into the host cell DNA. The patient cell now contains the gene that the patient needs. The cells that have been modified are then returned to the patient's bloodstream. Inside the patient's body, these cells reproduce, passing the gene onto their daughter cells. These cells are capable of expressing the gene and producing the protein that the patient needs to combat the disease. So there you go. That's gene therapy. This has been How Science Works. See you next time.